Hello. So we continue our survey on fungi module. Today's agenda will be to discuss penicillium. Before that, let us go through the usual procedure. Under zygomycota, we will be seeing rhizopus and mucor, which we have already completed. Then under ascomycota, we have these many life cycles: Saccharomyces, Aspergillus, Penicillium, Neurospora, Phaeacia, and Claviceps. Out of these, Saccharomyces and Aspergillus is already done. Penicillium will be completed today. So Neurospora, Phaeacia, and Claviceps remains. Now, under Basidiomycota, we have to see Vaccinia, Agaricus, and Ustilago. Then under fungi-like organism, under the phylum Oomycota, we have to see the life cycle of Phytophthora and Albugo. Then we will be seeing lichens, mycorrhiza, fungal toxins, and some other general topics. So before coming to penicillium, just understand that there are two types of fungi. One is unicellular fungi called yeast. Then there is multicellular fungus called mold. Under mold, we are to explore Rhizopus, Aspergillus, and Penicillium. And last day we have completed Aspergillus. Just see, there is a very visible difference between Aspergillus and Penicillium. In Aspergillus, we can see that the tip of the conidiophore is like a globose vesicle, and in the case of Penicillium, that is missing. We will come to these details in the upcoming slides. Penicillium is a saprophytic fungi just like any other fungi, and it is commonly known as blue or green mold or blue-green mold. For example, Penicillium notatum is a perfect example. Why it is called a blue-green mold, I will show you. This is why. When it is infecting one particular organic matter, you can see that it will be giving a bluish-green texture. You can see that there are three different types of mold. If Rhizopus is infecting the bread, then that will be giving a blackish appearance. So that's why it is called bread mold or black mold. In the case of Penicillium, it will be giving you a blue-green appearance. Now, and it is present in dead and decaying organic matter. Around 136 species are found throughout the world. And the wonder drug or penicillium as an antibacterial compound was extracted by Sir Alexander Fleming in 1929 from Penicillium species. They are also economically important for the production of cheese, organic acids, antibiotics, etc. Then under the systematic position, we have to know that it belongs to domain Eukaryota. Then kingdom fungi or mycota, phylum Ascomycota, obviously, class Uroshiomycetes, order Uroshiales, family Trichocomaceae, genus Penicillium. Okay. Now coming to the vegetative structure. Under the vegetative structure, we know that there is a vegetative mycelium. It will be multicellular. Okay, you can see multiple cells separated by septa, and there are some species where conditions like multinucleate condition available. Then in some species there will be each cell will be uninucleate. So here as an example for illustration purposes, we have taken a species with uninucleate cell in the filament. Okay. Now what will happen to the thallus structure during the time of asexual reproduction? That we will see in the part of asexual reproduction. This kind of a structure will be formed. I will show you in that part. Then remaining thing is there. You can pause the video and take the note. Okay. Now coming to vegetative structure, vegetative body is mycelial. It is profusely branched with septate hyphae. Each septum has a central pore. Okay, each septum has a central pore, and in this each cell, within each cell, you can see there is a nucleus, and this dark colored dots that you are seeing is the oil globule, which is the storage product. So coming to the remaining explanation, the reserved food is present in the form of oil globules, and you can see that the penicillium means painter's brush. Why? It is due to the presence of chains of conidia. Okay, during asexual reproduction, conidia will be formed in chains, and it will be appearing as a brush terminally on the mycelia because of this. Okay. Now coming to the reproduction part, it shows vegetative, asexual, and sexual reproduction. Vegetative multiplication or vegetative reproduction due to the fragmentation, accidental breaking of the vegetative mycelium. You can see like individual mycelium being formed. Okay, this is a microscopic view of the penicillium colony. If I am zooming in, you can get a mycelium like this, very profusely branched, and if I am again zooming in, you can see a structure with a uninucleate or multinucleate like thalli, okay, or mycelium. Asexual reproduction. During the time of asexual reproduction, one particular cell from the vegetative mycelium that will be arising, growing in an erect fashion. Okay, since the species we have taken is uninucleate, this one particular nuclei will be moving towards the conidiophore. Okay, So the conidiophore has arisen. From the tip of conidiophore, you can see that there is sub-branching in the form of ramus and medullae. On the top of each ramus, there will be 2 medullae. Okay. Then at the tip of the medullae, you can see sterigmata or phyallid, which are flask-shaped cells. On the tip of phyallid, you can see conidium, which is arising in chains. Okay. How this conidium is arising, we have seen in yesterday's aspergillus, same procedure, but I will show you again. So the entire broadly oval phyallid tip. So phyallid was a flask-shaped structure or oval-shaped structure. From the tip of this particular phyallid, cytoplasm arises or accumulates at the towards the tip area, and the nuclei divides. Okay. So once the cytoplasm pushes out the phyallid tip, it will be becoming a tubular or tapering structure. And once the nuclei has divided, one nuclei moves towards the tip. It is within this tip there is a uninuclear conidia which is going to be formed. Okay. So once the nuclei has reached, the nuclei will be accumulating a small amount of cytoplasm or protoplasm around itself, and that will be a septum will be cut here, and that will be separating it from the mother thalli. But there is a connection between the parent body and this particular conidium till it is formed through an outer wall. Okay, outer conidial wall. Okay. It is the inner conidial wall which is separated through a septum, but the outer conidial wall is still connected to the parent body till it is like breaking apart from the parent body. So this cytoplasmic pore connection will be present in the case of each conidia. So once the first conidia is formed, there will be again pushing of the cytoplasm towards the tip, and there will be one nuclear division through mitotic division. Then there will be accumulation of protoplasm around the nuclei, and a inner conidial wall will be cutting off a septum, and the first one will be pushed towards the front, and second one will be coming like this, then third one like this. So younger nuclei or younger conidia will be towards the bottom, and older mature conidia will be towards the tip. Okay, so this arrangement is known as basipetal arrangement. So like this is the thing that is happening. So the explanation is here now. So after the formation of this conidia, when the older conidia, the tip which is completely mature, and that will be releasing, and that will be landing on a suitable substratum, and that will be forming new hypha that will be like continuing the cycle by forming the conidiophore and other structures. So this is the knot. So you can see that different species of conidia, different species of Penicillium are displaying different structures with minor variations. Here you can see in the C, you can see from this particular conidiophore tip, this is the fashion in which this is arising. Okay, you can see two structures. Okay, maybe you call this as ramus. From the ramus there is directly phyallid formation, but here you can see that there is ramus on the tip, not here. Let us see. Yeah, no, actually all has shown this like one particular structure is missing here actually. This particular yeah, 1 2, if it is, yeah, let us see this again. Okay, this is ramus. So from the conidiophore tip there must be a ramus formation. From the ramus formation medullae has to arise. Okay, from the conidiophore tip there must be a ramus formation. From this there has to be what? Medullae. From there has to be then there has to be this particular phyallid formation. Here you can see that. Okay, this is the ramus, then this is the medullae, then there is the phyallid. But here that phyallid is missing, or this like medullae or ramus, like either one is missing. Okay, so these are the difference in different species. Okay, so asexual reproduction knot, you can take it here. I have given everything in detail. I have already explained all these things. Now, sexual reproduction. Sexual reproduction is also very simple. Just like in aspergillus, you can see that one particular vegetative mycelium, instead of one particular cell from the vegetative mycelium, instead of forming a conidiophore, what it will be forming? It will be forming a uninucleate ascogonium. You can see it's a uninucleate ascogonium because the cell will be uninucleate, and that particular nuclei has entered into this particular ascogonium. As it proceeds, as it matures, as it proceeds towards maturity, these particular nuclei will be dividing mitotically into around 64 nuclei. Okay, around 32 to 64 nuclei can be there in one particular ascogonium, and all will be haploid. Why? Because each cell had haploid nuclei in the vegetative mycelium. So that haploid nuclei entered the ascogonium, and that undergo mitotic division to form around 64 haploid nuclei. So beside this ascogonial hyphae, there will be another hyphae which will be forming similar growth towards the top, that is an antheridial hyphae. Okay, and this antheridia will be coiling around this particular ascogonium, and you have to observe one thing, that the antheridium is also uninucleate, but we are not showing the division of antheridial nuclei. So in the case of Aspergillus also, we have seen that in most of the cases the antheridium is not reaching the ascogonium, and only the ascogonial nuclei are pairing up to form the successive formation of ascospores. Here also that thing happens. Okay, the antheridial, though the antheridium fuses with the trichogyne of the particular ascogonium, this particular nuclear content or male nuclei is not getting transferred in most of the cases in the case of Penicillium. So this blue nuclei, after this we are not going to show this blue nuclei again, okay, because this male nuclei is not getting transferred to the ascogonium. So what will happen is that next thing will be, we are moving towards the crozier formation. So how it will happen? Actually the crozier formation as such is not happening, but it will be forming the remaining steps, like for the ascus formation and ascospore formation, that will be happening. So in this case we can see that after the fusion of the antheridial hyphae and ascogonial hyphae, not the fusion of nuclei, just the fusion of hyphae, you can see that the male nuclei is not getting transferred to the ascogonium. So what will happen, because of the not availability of male nuclei, because ascogonium is having many nuclei, here you are having only one antheridial nuclei. Even if it is ended, is nothing going to happen, because there is one, and here it is 64 nuclei. So what will happen, these particular ascogonial nuclei will be pairing up like this, 1 line, 2 line, 3 line, it will be pairing like this. Then after pairing up, there will be segmentation. Okay, now many cells are created. From each cell, I am taking this particular cell, from each cell there will be a generation of ascogenous hyphae. Okay, this will be going to form the ascospore mother cell. That's why it is called ascogenous hyphae. From this cell also an ascogenous hyphae, and I am taking this particular new hyphae. Okay, this is the particular new hyphae. The tip of the ascogenous hyphae, which is having a dicatotic condition, that will be going to form the ascus mother cell by turning into a globose structure. Okay, now at this stage, these both two nuclei fuses. Now it has reached a diploid zygotic condition. From this particular diploid nuclei will be dividing, one meiotic division and one mitosis division. So that will be forming around 8 cells, 8 nuclei, and each nuclei will be accumulating a cytoplasm around itself, and it will be forming ascospores. So this is just one asci. This is one asci at the tip of this particular ascogenous hyphae. Here also there will be one asci, and here also there will be one ascogenous hyphae and one particular ascus mother cell. So there will be an n number of ascus ascus mother cell and asci created. So that entire structure will be covered by sterile hyphae, and it will be forming a globose or oval shaped structure called the cleistothecium. This is the ascocarp of this Penicillium ascomycota. So what will happen then at the time of maturity, ascus or cleistothecium wall and ascus wall will be broken apart, and ascospores will be released outside, and that will be forming germinating form a germination tube or germ tube, which will be maturing into the vegetative mycelium. You can see simple arrangement given here. Okay, without much elaboration, but we have to know this elaboration. It can be a separate question. The formation of ascus mother cell and the cleistothecium. Okay, formation of cleistothecium in Penicillium can be a separate question. So you can proceed to the yes, similar is this is a colored image which is drawn. This is given in textbook, same thing only. Then this is, I have taken this from another source, but there is fundamental mistakes in this particular image, just for you to show. Just identify and comment in the comment section what is the fundamental mistake in this particular image, in the usage of color and the number of spores, etc. So you can pause the video and take the notes. All the notes I have explained in detail. So with this we are winding up Penicillium. In the upcoming lectures we will be seeing all the details regarding the next fungi genus. Till then, bye. Take care.